Ah, right. To introduce Bill Cresko, who needs no introduction. I'm looking forward to seeing what our good colleague has to say.
Yay. For the introduction, can everybody in the audience hear me? Good. Okay, folks on Zoom, can you hear me?
Yes.
Excellent. A little bit about some of our old. But before I get started, I actually want to up on top there. And so I'll point that out at different. This is the first. All right, so with it might be this.
Ask them, how's that?
How's this now?
Can you hear me, Bill?
I can, Patrick.
Yes,
Phillip.
so we can hear you occasionally. And then when you turn your head, it seems to go away, or you're moving away from a receiver, or the battery is bad if that's a remote thing. It's kind of cutting in and out, and we're mostly hearing less than 10%.
I think so.
Or here, actually, I just do this here.
Yep. Might want to actually try just the computer. Not working.
You could try it. Is that this one? Yes.
Yeah, try that.
Well, you'll have to stand closer.
Yeah, so I'll try to stand right here. So I'm just using the computer microphone right now. So Zoom folks, how's this sound?
That's fine the way that you're oriented right now. So we'll see.
Okay. I will try not to move away from the computer. All right, I'll see what I can do. All right, shall we keep going? All right. Thanks. Sorry for the brief interlude right there. Okay, so really, this is one of the big questions that, like I said, is deceptively simple, but why are organisms the way that they are? Well, one answer, and of course, it's not everything, is evolution, right? So organisms evolve over time, but the real deep question is, but how? How is that actually occurring? Well, to actually go into this in a little more detail and show you how my thinking's changed over time, we have to just define some terms here. So first off, if we think about evolution in one definition, it's largely population genetic. It's a change in allele frequencies over time that oftentimes leads to the evolution of to phenotypic divergence. When we think about phenotypes, and those could be phenotypes that are morphological, life history, behavioral, molecular systems, an adaptation is a phenotype that performs a function and has evolved to its present form via natural selection to do so. So if we take that, and of course, there's slight variations on these points. So if we think about evolvability, then that's the capacity and directionality of a system for future evolution. So when we think about evolvability, we can in some ways think about it as almost as a adaptation or as a property of a system. And when we think about adaptive evolvability, that's the present state of a system has evolved via natural selection in some way that influences future evolution. So if we think about this, this is kind of an important distinction, because if we think about questions about the genetics of adaptation, one of the core models we have in our mind that's been around for a long time goes back to Fisher and a number of other thinkers in terms of a geometric model where there's a sequential steps of sequential fixation of small effect mutations. But of course, many workers and the researchers in the field have pointed out that evolution may happen that way, but doesn't need to. And is it important that evolution occurs in space and time? And I think the answer that many of us know is yes. And I'm actually going to show you from Threespine Stickleback today how I think this really matters for the way that we think about evolution in that system. And I'll tell you more about that in a second. So what are some questions that really matter in the context of evolvability? Well, one of these is, really, if we think about sex and recombination as a system, why do they exist and how do they evolve through time? So if we think about the evolution of modifier loci that might actually affect rates of recombination within and among different populations. And Dr. Singh had told you all about that recently, and work in her laboratory has actually led the way in many of these questions. Questions of the evolution of sexual reproduction in Patrick Phillips' lab and many others. Why are there co-adapted gene complexes? So why do we sometimes see loci that actually show relatively low or no recombination together in parts of the genome? If we think about that, we actually have to think about that in the context of the system and the evolvability of that system. Why are some organismal traits modular, meaning that their mapping from genotype to phenotype actually has a certain organization to it that might actually mean that there's certain patterns of variation that can actually be explored in phenotypic space? And why are some robust so that you can actually have mutations? So how did that robustness evolve through time? And why do organisms exhibit phenotypic plasticity? So the capacity to consistently exhibit the same phenotype, consistently exhibit phenotypes of the same genotype under environmental variation. So we explore this in a variety of ways in my laboratory. And one of these is at really deep evolutionary scales. And this is some researchers in the evolvability have actually asked this question from the standpoint, are there very low frequency unique changes that have actually happened in certain lineages that set off new directions for the innovation of novelty? So this is actually an X-radia microscope scan of the head of a sea dragon. So a fair amount of the work in my laboratory is focused on the evolution of synaphids, sea dragons, seahorses, pipefish, and their relatives. And so this scan right there shows you something amazing, is the highly derived head and jaw structures of these organisms. Members of my laboratory have actually shown that concurrently there are some pretty radical changes in deeply conserved genetic systems in synaphids, including regulatory elements in Hox genes, which are important for anterior posterior patterning, as well as loss of members of fibroblasts, FGFs, or fibroblast growth factors in the lab. I won't tell you anything about that today, but it's actually along those lines. What are those low frequency events that happen very infrequently that set up new directions for innovations? Some of the work that I've contributed to in the past, and it actually has a lot of its genesis here at the University of Oregon, is how do modular gene regulation actually arise and evolve through time? So this is just a little schematic from a paper a number of years ago, where if we think about a single gene that actually has a single function, or we can call it a subfunction, how does that subfunction, if the gene has multiple different roles in different tissues, how does that actually evolve through time, through the divergence of those subfunctions and then eventual partitioning between different gene duplicates? And if you think about that evolution, that leads to a change in that system that will have strong influences on subsequent evolutionary change in that system. But today I want to focus on another branch of my laboratory that's largely, I'm thinking about the population and quantitative genetics of organisms in natural systems that we can observe in nature today. And I'm going to focus on a couple questions that we've been struggling with, which is, do developmental genetic systems in natural populations exhibit adaptive evolvability? Of course, I'm not going to answer this question generally. I'm going to use one specific case study using the small fish Threespine stickleback that I'll tell you more about. And does the evolution of adaptive evolvability, if it exists, depend upon the ecology of the organism? I'm going to jump ahead and say that I think the data from our and other laboratories studying stickleback strongly argue that both of these things are actually true. There's much more work to be done. And at the end, I'll come back to this. And it really matters for that question of why are organisms the way they are? To really answer that question, I think we need to think about this from that context. So before I get into the data for this, I just want to dig a little bit into the organism that we've been studying. And I'm going to stop right there and just ask the Zoom folks, how's it been so far? Have you been able to hear me up to this point?
It sounds great.
Awesome. Good. This little guy, Threespine stickleback, Gasterosteus aculeatus. So this small fish has been a workhorse for studies in ecology, behavioral biology, and evolution for a number of years. Part of the reason is because of its unique life history. So this Threespine stickleback and nine-spine stickleback have a common ancestor between 15 to 25 million years ago. And over the last 15 million years or so, stickleback that are ancestrally oceanic have invaded freshwater habitats over and over again. And as the glacial cycles have actually proceeded on the planet, there's actually been an opening up and a closing of new freshwater habitats. And so this process has happened repeatedly. And when this has occurred, there have been changes in a number of different aspects of those fish. So changes in size, shape, coloration. This is just a set of images of pairs of oceanic and freshwater stickleback that differ from one another in their nuptial coloration. So I'm just going to look at my screen. I'm not going to turn that way to point at it, but I think you can see, oh, there somewhere, there are different colored stickleback. One of the aspects that is most notable is that if you actually look at their external bone phenotypes and internal bone phenotypes, but their defensive armors, the presence of lateral plates, those series of red things that go down their side, and the pelvic structures that are on the stomach, which are highly derived pelvic fin. These are stickleback that have been killed, cleared, and stained, so you can actually clearly see the bones. And these are general morphs that in terms of the presence or absence of the lateral plates, and presence or absence of the pelvic structure. So if you look at those closely, you can see plates, no plates, presence or absence of the pelvic structure. Those are independent traits that actually come and go. And work from Mike Bell and colleagues has shown that this has been happening. The evolution of the loss of this armor and change of this armor has been occurring for millions of years. So there's a number of fossil beds in Nevada that Mike and colleagues have looked at very fine scale, can actually show changes in these phenotypes. So this has been happening repeatedly in numerous different locations throughout the Northern Hemisphere and coastal regions in millions of different lakes, literally millions throughout Russia, Europe, North America, and has been happening for millions of years. Speaking of a long time, this is me a long time ago in Alaska with stickle nets that you can make yourself with duct tape and sticks and nets and a floating bucket and go out there. So when I was a PhD student, I actually started working with Susan. Susan's a behavioral ecologist, ethologist was, and actually made some fundamental contributions to our understanding, the world's understanding of how behaviors can evolve geographically in many of the same way as other traits. Half of my thesis was actually working on how changes in mating behavior across different populations could actually lead to the evolution of reproductive isolation, prezygotic reproductive isolation. But I also had this strong interest in how changes at the genomic level could actually be occurring across different populations in Alaska that we were seeing or evolving many of these phenotypes at the same time. So this was in the late 90s. So the work that I was doing in population genetics was involving microsatellite loci with P32, loading gels, Andy's, yeah. Loading that, and every single time one of my hairs from my dog ended up in there and I had to pour the thing again. And I got really frustrated because we could see things like, yes, there's a general pattern of population genetic diversity, so that most of the diversity in freshwater populations that existed in oceanic populations existed in freshwater populations, but we couldn't see which genomic regions were changing and why. And I guess one of the points that I would make from this talk is harness that frustration and use it to actually do something again in the future, just because it bugs you that you weren't able to answer it. And I'll tell you what we did here in a second. So the other thing that it actually made me frustrated is that I really wanted to know the underlying genetic and genomic basis of many of these traits. And where I was doing my PhD at Clark University wasn't the best place to do that. One of the best places in the world to do that, studying fish developmental genetics, was right here at the University of Oregon. So I reached out to colleagues, John Postlewaite, Chuck Kimmel, and was able to move the Stickleback system here and work with them. And that actually set up the working with many of the other folks here. And it was one of the best decisions of my life. But it was extremely difficult. I'll say the easiest thing would have been just to move into another evolution lab. The harder thing was to actually move into zebrafish developmental genetics, but it paid off. So I think that's another thing that I think is important. So a lot of the early work that we and others in the laboratory, in the world, in particular, David Kingsley's lab, Katie Peichel, Craig Miller, Frank Chan, a number of different researchers were studying different traits and have been since then in Stickleback. And there's several things that actually showed up. One is that there were large effect quantitative trait loci that were identified using quantitative trait loci mapping in the laboratory. This was actually contrary. So this 2004 paper, if you look in there, you'll see all these quantitative complementation crosses that I did because I was convinced that these were not going to be the same loci underlying the repeated evolution of these traits. And it was all, at that time, a waste. I'm not sure now that it was a waste, but at that time I was like, oh, it doesn't really matter. I will point out, though, that some of those quantitative complementation crosses for the pelvic structure did show that there were different loci segregating even at that time, but not for the lateral plate. And the same genomic regions, whether this mapping was done on British Columbia populations, those in Europe or in Alaska, went to the same regions of the genome, which frankly just shocked me and many of us. So we're finding that it looked like the same traits had at least an underlying parallel genomic basis. So this is really old work, so I'm just highlighting because it actually sets up the work for the future. And this really drove home the question as I opened my laboratory: what is the genomic basis of that, the pattern of genomic differentiation across Stickleback adapting to these different habitats? And that really brought back some of the time that I was spending my PhD where I just didn't have enough data. So I was convinced that after I opened my lab here, I was going to blow all of my startup on making SNP microarrays. So another thing that happened when I first joined the group here in Oregon in the mid-2000s was Karen Gilliman at that time was running a faculty group where we would all get together and pitch crazy research ideas. And I think it was a really great idea for a variety of ways, really enjoyed it. And one of the times that I was there, I pitched this idea of SNP microarrays to do genomic scans. And my colleague, Eric Johnson, said, hey, we started toying around with this idea of actually using microarrays in the laboratory and this thing called restriction digestion associated, we didn't have a name for it at that point. Why don't we give that a try? And so Eric and I started a collaboration that led to the development of RAD sequencing. A lot of the data that I'll show you today actually comes from that. And really, it's actually taken off from there. So that necessity of what I wanted to do and that bug that I had be in my bonnet actually led to collaborations that, again, were challenging and uncomfortable in good ways, but I think in the long run were really beneficial. And this actually led us to then go back to some of the same populations of Stickleback in Alaska that involve those low lateral plate and pelvic phenotypes and ask, well, what has happened across the genome in 13,000 years? And this is a picture from a paper from 2010, and this is Paul Hohenlohe and Susie Bassam, who were the push forward the work on this paper. And this is a picture that I found. This is for the Barber Laboratory. They're looking for the Barber Laboratory in Edinburgh. So this was a workshop that we did. Fortunately, we were 10 years too early looking for them. So you'll notice Paul and Susie were a little younger there. But one of the things we identified is that when we looked across 3 independently evolved freshwater populations, the same genomic regions were identified. So each one of these plots shows you from left to right the Stickleback chromosomes in alternating colors, and the y-axis is FST, which is the relative amount of divergence between ocean and freshwater. So low means very low divergence, high peaks mean very high divergence. Every one of those yellow lines actually indicates regions in common that were divergence across those populations. A couple of years later, work out of the Kingsley lab actually showed that this was not just a pattern that was local to Alaska, but was very global, so that the same genomic regions were evolving over and over again. And this led us to a question of, well, if that's happening, and it's happening over 13,000 years, how fast could it happen? And this is work that brought in our collaborator, Frank von Hippel from the University of Alaska. His graduate student was co-advised by me, Emily Lesek. Julian Katchen was a long-term collaborator in the lab, developed the Stack software platform for the analysis of RADSeq data. And Mary Sherbik, who was an undergraduate student and Alaska native, who actually was a huge contributor on this project. So we had a couple papers that was focused on a set of islands that were uplifted on March 27th, 1964 at 5:09 p.m. Exactly, we know when it was. Part of this is because this was one of the most significant earthquakes ever to have actually happened on the Earth. So it was 9.2 on the Richter scale. And just to give a comparison, I think the Loma Prieta earthquake was somewhere around 6 on the Richter scale. So what is this, 1,000 times stronger than that? What that meant was that there were significant upheaval of a number of different areas. This is, sorry, I jumped over this, but this is downtown Anchorage. People went into stores, the earthquake happened, came out, and there were a story above where they were previously when they went in there. So major changes. And one of the changes is that new land was created around the edge of many islands in the Gulf of Alaska. And many of these islands were not inhabited, but actually were known and mapped because of, well, we were in the middle of the Cold War, so they're actually utilized for a variety of different reasons. And one of those islands was Middleton Island. So on the left is an image of the outline of Middleton Island before the earthquake and the uplift. And on the right is a picture from 2008. And you can clearly see that fringe around the edge of the lake. And what Frank and his laboratory had actually identified is that in those little ponds around the edge of the lake, they were no longer saltwater, they were freshwater, and in those appeared to be freshwater looking stickleback, even though there were no previous freshwater populations on the island. There was one, we did some work to actually show that none of those little ponds around the fringe were actually derived from that existing freshwater population of stickleback on the island. A lot of work in the field. This is largely Emily and colleagues going there. And the upshot is that when we looked at those islands, not only Middleton, but Danger and Montague Islands and many others, there were numerous different geographic locations that had independent evolution from oceanic to freshwater within 50 years. And we could date this to that time period. So going from the middle to outwards on this circle here, I've changed that plot that actually showed the peaks up and down to a heat map, just to give you a little better sense of this, where green is actually a low value of FST and red is a high value of FST. So starting from the middle, that's just a comparison of ocean population to ocean population between islands. So as expected, there's very little or no divergence between those populations. Moving outwards, and you can just see the second one is Middleton freshwater versus Middleton marine. The next one are sympatric ecotypes that exist in some of the lakes, the little ponds that were around the edge of the lakes. There's sort of morphs that look freshwater and some that look oceanic. And then moving further out, it's a completely different island, Montague Island, and then Danger Island, and so on and so forth. The details don't matter as much as compared to the big trends, which is, as we'd seen previously, a large proportion of the linkage groups in the genome, those are identified by the 1 through 21 around the outside, showed very little differentiation. As a matter of fact, in comparison to what we found in those older 13,000-year-old populations, when you look at these linkage groups, there's statistically no divergence. So if you were to ask what's the population structure of these stickleback in these populations, in those linkage groups, the answer is there is none. But if you look at those genomic regions that we and others had identified previously, there's significant and consistent divergence between those. So very rapidly, there's changes in allele frequencies that were actually occurring. And this was happening over very short time periods. And you'll notice that it's very block-like, right? So you actually have chunks of the genome, especially on linkage group 4 and linkage group 7 as you're moving clockwise through there. But you also notice on linkage group 11 and linkage group 21, that's pretty significant because there are in known inversions on linkage groups, in particular 11 and 21. I'll show you 21 here in a second, but also suspected at least some structural variants that exist in some other parts of the genome as well too. Oh, sorry, linkage group. I'm going to try not to. If you look in around 7 p.m., linkage group 11, where that arrow is actually one inversion, and then linkage group 21 is another inversion up there. So there's another one on 1 as well too, but I just highlighted those ones in 1 and 21. So this actually led to another question in the laboratory, and it's another theme I'm hoping to, this progressive nature of science, right? Every answered question then leads to a next question is, and this really fascinated Tom Nelson, the graduate student in the laboratory, along with all of this is, well, if this is the case, what's going on at a deeper evolutionary time for these genomic regions? What is their age? You know, are they young or are they old? We would actually predict that what's happening in 50 years is the repeated evolution from standing genetic variation. But if that's the case, then what's the nature of it? One thing I forgot to mention a second ago, very recent work in populations of stickleback that have actually been seeded in lakes in Alaska has actually shown that change in these genomic regions actually happens on the order of a decade. So it's very fast. So I just wanted to point that, I forgot to mention that. So this is work that we published a couple of years ago that Tom really led the way on, and he wanted to take a coalescent analysis approach. So instead of thinking about allele frequencies, let's think about the sequences of genomic regions and what that tells us about those ages of those genomic regions. And what was important about this is that we didn't want to do it just in one place of the genome or another because the different parts of the genome had very different histories. So he wanted to come up with a way to actually do coalescent analyses on a genome-wide scale. So one way to do that would be to resequence entire genomes, assemble those, align those, and do that. But that was prohibitively expensive then and is now. We didn't actually need to really do that because what Tom said is, well, if we can actually go through and sample the genome at a high enough density using rad markers like we've done before, we can estimate the evolutionary history of these sequences and estimate the time to the most recent common ancestor for each genomic region and compare them across the genome to see how they compare and how they compare with where the genomic regions show the greatest divergences compared to neutrality. So what Tom did is he developed an approach where he really kind of took the rad sequencing and focused it on the cut sites in the genome, which is showing you from the top there, and was able to phase the paired end reads on both sides of the cut site and then phase across those as well too. And in doing so, we were able to get about 700 base pair fragments, which we showed, that Tom showed very well, that with the levels of genetic diversity in stickleback was sufficient for us to get pretty good estimates and a locus by locus basis for the coalescent history, which is pretty cool. So then that allowed us to go through and recreate and estimate the coalescent history at each of those genomic regions, again, for those same freshwater populations we studied previously as compared to the oceanic populations. And summarizing a huge amount of work that I just want to highlight here is that when we looked at that inversion on linkage group 21, so this is the one, Andy, that I pointed out that was on 21, which is that big block, which is known inversion of 2 megabases in length. The time to the most recent common ancestor of those peaks that are on the left there is much deeper than one would expect just from the neutral expectations from the entire genome, which is the dotted line. Whereas when you move outside the inversion, it's about that expectation. Furthermore, you'll notice down on the bottom there, I think, if you can see on the screen, little blue boxes and little red boxes, they were distributed between oceanic and freshwater forms. So you actually had alleles that were ancestral for oceanic, that were consistent for oceanic, alleles for freshwater, relatively short coalescence times for each of those, but very deep between them. So this was really pretty intriguing. To summarize this, and I'm going to turn now on the side here, an evolutionary history for different populations of rabbit slough, bear paw, bootleg, which is BPBL for bear paw, bootleg, rabbit slough. And then we needed to root this. So Tom actually generated the first genome for the 9-spine stickleback and used that as the outgroup. And that way we were able to link this, identify the common ancestor about 15 million years ago. But in doing so, it might mean that it's more like 20 million, 25 million years ago. It doesn't really matter too much because we were able to use the relative amounts of coalescence times within and among the morphs within Threespine as compared to the 9-spine. And what Tom found was pretty striking. But if you looked across the Threespine Stickleback genome, that more light distribution there shows the time to the most recent common ancestor, so TMRCA, for the genome-wide average. But if we looked in regions that have been consistently identified as divergent between marine and freshwater, what we see is that it's decidedly shifted to the left. So many of these genomic regions were actually many millions of years older. So the time the most recent common ancestor, the mean for that distribution is somewhere around 6 million years. But one of the things you'll notice is there's a long tail there that actually some of those predate the divergence between 9-spine and 3-spine. And more recent work from Juha Marila's lab has shown that probably some of the variation is actually ancestral to the common ancestor between 9-spine and 3-spine, and it's actually been reused in both populations for repeated, or both species for repeated adaptation, which is way cool. But why? How is this happening? How are this evolution happening so quickly, so fast? And also using these same genome, why aren't they being recombined and actually divided? And why do you actually see this signature of molecular evolutionary history? Well, one of the things that we've proposed is that what's evolved concurrently is the genomic architecture of the haplotypes. So that what we might actually see is that the local rates of recombination vary across the genome, but also where those genomes are found. And so what Tom and actually 2 undergraduates in the lab, John Crandall and Kate Tuarta, worked on with others in our group, was to be able to take advantage of the fact that even though we didn't have inbred lines, we could use what are called pseudo test crosses by combining individuals that were genetically diverse and then looking at markers that actually had the pattern that you might see in inbred lines, and then using those to map male and female genomes to actually make male and female maps. So basically you're saying, all right, in those progeny, you're looking for situations where you actually have heterozygotes. You can map those versus homozygotes, which ones are specific to males, which ones are specific to females. Because what I'd say is if you'd like to learn more about this, I can tell you it works. I'd be happy to talk to you about it later on, but you can make pretty high density maps. And so what we did is we actually took a set of freshwater and marine fish and mapped them, and then also crossed them together. So we were mapping hybrids. Now, remember, this is so you have 2 genomes together in a single F1 individual from marine and from freshwater, and then asked about the recombination rate in that male or that female, because what you're basically doing is asking about the recombination rate in those individuals. And what we did is asked about certain individuals, and in this case, we can just show how many progeny we had for each one. You know, not a huge number, but good enough to actually be able to have decent local estimates for recombination rates and good density of markers across the genome. So these are pretty high density maps. And so what I'm going to show you here in a second is what does the map look like for the genetic map, which is the recombination rate for within an individual that has 2 freshwater genomes, 2 marine genomes, and an individual that has a freshwater and a marine genome. And our prediction was that these would look different. So we'll go back to that inversion on linkage group 21, and we'll have those little pictures down the bottom, freshwater, marine, hybrid. What we actually see is that when you look in the freshwater, there's not really a restriction of the genetic map as compared to the physical map on the top, or the map of the genome. Pretty much those orange lines that designate the region of greater divergence between freshwater and oceanic populations are straight. They cross, but it's about the same distance. It's what you'd expect because it's an inversion. What happens in the hybrid? It goes to a point, just like you'd expect. That's pretty cool. But what about another region of the genome that has been identified as being involved in the lateral plate reduction, in a variety of other traits, seems to actually evolve over and over again, but we don't have known inversions on that chromosome. Here's the physical map. Again, blue is background, orange is divergent. What we actually see is that there is similar constriction down to a smaller region of the genome, but it's not just specific to the hybrid. It actually happens in the freshwater map and the oceanic map as well too. For some reason, there's a restriction of recombination that's actually occurring here. And I'm not going to show you all the other maps, but this is happening in most of the places across the genome. And I can summarize it here, though, which is that if we think about the background as being the physical map, and if we think about 20% of the genome, and the physical map genome is showing these patterns of divergence between ocean and freshwater, what percentage of the map, genetic map, is that for each one of these? Well, it's significantly reduced in all three of them. There's a reduction in rates of recombination in those regions that co-occurs with the divergence between freshwater and ocean, which is pretty striking. All right, so that's the end of that story. I just wanted to kind of tie this back into something that another line in the lab, a dead end that actually led us to a little bit of a breakthrough, and then finishing out. Depending on how things go, I might just skip the a section at the end. But we really want to know how really modular is the Stickleback genotype to phenotype map that results from QTL mapping in the laboratory. Kind of bugged me a little bit, and the fact that we actually see this variation in recombination rate also let me know that we really need to dig into this a little bit more, and not just accept that there's major loci that are contributing these traits. Maybe they're linked minor loci that appear to be major loci. So what Kristen Allagood and Mark Curry, this is a picture of Mark when he was a young man in the laboratory. So I like this picture, although you don't look much different right now. So we did the naive wrong thing, I will say, which is, hey, let's just go to Middleton Island and go through and they show phenotypic variation. We should be able to just control for population structure and map the phenotypes using GEMMA. No problem, should be fine, Kristen. As Dr. Singh has told many of you numerous times, linkage and linkage disequilibrium are not the same thing. And linkage disequilibrium can occur for reasons that have to do with population structure, mutation rates on different backgrounds, and the action of natural selection, and which is very much what's happened in Middleton Island. So what I'm showing you here is a pattern of R squared, which is a measure of the non-random association of genomic regions with respect to one another within linkage groups and across linkage groups. That's that big pie in the middle there. The triangles on the outside are within the freshwater populations in Middleton and the oceanic populations. You see very little red, which is very little linkage disequilibrium in each of those populations. But when you look between them, you see significant amounts. And, in retrospect, this makes total sense, right? Because you've got these chunks of the genome that are actually evolving very quickly. But maybe we can control for that population structure. I think that's the important thing is, remember what I told you before, is that when we look in parts of the linkage group, the genome, like linkage groups 15, 16, 17, there is no population structure, so there's nothing to control for. But when you control for it in those regions of the genome that are actually the regions that are under selection, that are presumably under selection because they're linked to phenotypes, what are you controlling for? The things you actually care about. And that's exactly right. So if you actually use on the top there, that's forget about what A and C are. It matters, but the important thing is when you do the Manhattan plot on the right, looking for an association between phenotype and genomic regions, and you control for population structure using a region of the genome that doesn't have structure, you see linkage with all these different regions of the genome. But even when you actually try to control using, for example, linkage group 4, you don't get rid of them, but you'll notice that there's still associations between numerous genomic regions. And in this case, I forgot to mention, we're actually mapping the lateral plate phenotype, which we know is a single locus on linkage group 4. We still see these associations in different regions. So this wasn't going to work. So we decided to search the planet for the perfect mapping population. The reason that we really wanted to identify mapping populations is because you couldn't get enough recombination events occurring in the laboratory. We wanted to find a hybrid zone that showed recurring and repeated recombination. So we looked all over the world, and it took us to Springfield, Oregon. Didn't really do this. This was a little serendipitous, and this comes out of Mark Curry's work for his master's thesis, actually identifying the population structure of Stickleback throughout Oregon. And what Mark found is that when you actually look at the genetic signatures of the populations throughout the state of Oregon, especially in the McKenzie River Basin, the Willamette Basin, that On the left there, this is what Mark calls a subway diagram that created for this. Those little bars there represent a structure analysis that was done on these populations. What we actually find is that there's generally two different freshwater groupings in the lower reaches of the Willamette River and the different rivers that come off of there. And then a different freshwater up above. You see some hybridization in there. But in the Mackenzie River, you see something strange, which is that blue, which is actually oceanic genotypes of stickleback, which is like, what the hell's going on? Why are they in there? And the best thing that we could, excuse my language, what's going on there? The best thing we could actually come up with is that these were introduced marine stickleback into above the Lieberg Dam, presumably introduced when there was stocking that was going on, that actually has led to repeated recombination with the local freshwater populations. ODF and W say, no, this can't happen. There's no way, but there's the data. What's nice about this is that this riverbend population shows very little structure to it. So, meaning that the linkage disequilibrium that exists is primarily within the linkage groups, even though there's huge amounts of phenotypic variation across them. So if we go back and take that lateral plate presence absence phenotype and use this population to map it, we actually find just what we'd expect, a huge significant peak on linkage group 4. And what Kristen found is also, even though it looks small, if you look on linkage group 20, it's actually quite a significant peak that encompasses a gene that is a very logical interactor with the lateral plate phenotype as well too. It's in the Wnt beta-catenin pathway that makes total sense for the interaction with the ectodysplasin A gene that's in linkage group 4. So I just sort of wanted to finish up on that there because there's a lot more to come on this. So we really started a fair amount of work, and I'll say that the pandemic really got in the way, and we're picking it back up right now and really kind of cruising ahead on that. But looking at many different phenotypes. So how can we do this in a high throughput fashion for thousands of fish and many different phenotypes to ask about this modularity of the genotype to phenotype map? So if this interests you and this is something that you're interested in, I'd love to talk to you more about this. And then the last thing I wanted to finish up is another branch of research that's going on in the laboratory, which is asking us a different question, but it's related, but more from the gene regulatory network standpoint of things. What's the epigenetic landscape of rapid evolution in Threespine stickleback? So what's actually going on at the cellular level? So we can ask at the phenotypic level, genotype to phenotype map, but what's going on at the cellular level? And this is work that Susie is really, Bassam in the laboratory is really pushing forward, and that has input, even though Hope is doing work on a different project, the work that Hope's doing is really helping us move this forward. So I just wanted to give you a background why this is something that we think might be important. And this goes back to one of my favorite papers from our laboratory. This is actually from a paper in evolution that Katrina McGuigan, quantitative geneticist who spent time in our laboratory and Patrick Phillips' laboratory, we're really interested in understanding whether there was cryptic genetic variation in the oceanic stickleback. Could we actually take oceanic stickleback and raise them under freshwater conditions and see the exposure of phenotypic variation that implies underlying genetic variation and that has underlying genetic basis that would mean that there's genetic variation that's normally not seen, but then is exposed under certain environmental conditions. So what we decided to do is take a quantitative genetic approach using half sieve design, where we took 50 sires, and to each sire, we mated 2 females. So the individuals that when you move down, you've had sire 1, dam 1, all of those are full sibs from one another. We took 5 of those full sibs and raised them under a saltwater condition, 5 under an ocean condition. Then we took sire 1, dam 2, 5 under saltwater, 5 under freshwater, and did this a whole bunch of times. This was a huge amount of work, as Mark can attest to how long we had to go through and raise these fish up and then phenotype them. And what this allows you to do is partition not just the phenotypic variance, but the underlying genetic contributions, because the averages across the different males and those replicates give you an estimate of the genetic variation across males, and then the paired females give you an estimate as well too. So if we first start looking at the phenotypic distributions of the native environment, again, these were all oceanic stickleback, and native environment means saltwater environment. What we see is there's very little difference in the phenotypic distribution of those individuals across those families. So what this means is there just wasn't this huge increase in phenotypic variance. Things didn't just go haywire in freshwater, and you just have this huge amount of phenotypic variance. As a matter of fact, those distributions were not statistically significantly different from one another. So the trait here is overall body size. Sorry, I forgot to mention that. So this is overall body size. Good question that I forgot to say. But what happens if you look at the additive genetic variance? And this is nuts, right? This is crazy. When you actually raise those, when you look at those fish, remember, these are the same sieves, they're same estimate. When you look at them under the native high salt environment, there's no additive genetic variance. Heritability is 0. When you look at them under the low, the heritability goes to 0.4. Significant amounts of heritability that are only because the same developmental genetic networks are experiencing a different environment. How is that happening? So this really leads to a lot of the questions that we're pursuing in the laboratory right now using single cell approaches. So Hope is actually really pushing forward on the single cell approaches in the laboratory for her questions about what's going on in terms of the gene regulatory networks and the early development of synaphids in pipefish as compared to stickleback. But some of the work that Hope needed to do to begin with is have a basis of let's look at single cell approaches in stickleback. So what's a single cell approach? What this means is that instead of using whole organisms or tissues, modern genomic approaches allow us to go through and by cell basis, identify all of the genes that are actually being expressed in that cell, cluster those together and identify different cell types. As well, there's additional approaches that allow you to do this to not only look at the transcriptional landscape, but the chromatin landscape. And I'll talk to you a little bit about that in a second. So both of those can be done in bulk RNA-seq or single cell RNA-seq. And then there's an approach called ATAC-seq, and there's a variety of different approaches for this. But the ATAC-seq one is that we're really kind of pushing forward in the lab that Susie's optimizing. So what Hope needed to do first, though, is identify whether the stickleback genome was good enough to actually use for single cell RNA-seq, and it ends up that the 3' end of the 3' UTR of the gene, you actually needed to be able to have good annotations for the genes in the genome to be able to actually do single cell work. And it ended up that over half of the reads just did not map the stickleback genome. It wasn't good enough. So instead of stopping there, Hope said, all right, let's fix the genome. And so what she did is actually use an approach that she's called, is this scissor-seq? What do you call it now? Scissor-seq. Okay, scissor-seq. So the whole approach here, though, is to not use short read Illumina sequencing, but to use long read PacBio. And from that was able to go through and change the number of single cell RNA-seq reads from the Illumina that are mapping the stickleback genome very significantly. And there's additional comparisons in the paper if you'd like to read about them, but I'm just highlighting the fact that a single smart cell PacBio allowed Hope to actually go through and basically really improve the single cell RNA-seq analysis. So this is from a 70-hour post-fertilization stickleback, and each one of these things in that UMAP ordination are clusters of groups of cells that indicate different cell types. So in stickleback, this allowed us to actually allowed Hope to really identify cell types that lined up very nicely with what had been published previously in zebrafish. So that approach really helps Hope in terms of the work that she's doing in the laboratory to actually identify, well, what's different in those synaphid lineages in terms of early development of neural crest cells that are important for bone or blood cells or aspects of head and jaw development. But this actually allows us to go back in and ask this question using similar approaches in stickleback to say, well, what is actually happening at the transcriptional level and the epigenetic level when we expose stickleback to these divergent environments, and analyze at a bulk level and a single cell level. And we're making the prediction that we'll see is a coordinated response. So it's not just a general change in what parts of the genome are being utilized, but the same regions of the genome will move from a closed chromatin state to an open chromatin state under the influence of those different environmental conditions and correspond with the changes in differential expression. and that those regions are corresponding with those regions of the stickleback genome that have evolved repeatedly over millions and millions of years. If so, that fulfills a prediction of an adaptively evolvable system, which is a system that has evolved to its present state to actually then channel, in this case, inputs from the environment to produce phenotypes in ways that are consistent for the future. And one of the things I forgot to mention at the beginning, this is not teleological anyway. It's not like these systems know what's going to happen in the future. There's no gut. But you can think of this in terms of teleonomics. Systems actually can evolve to the point if there's regular experience of variation at a spatial or a temporal scale. All right, with that, I just want to finish up and just say, here's the overall conclusions. Where are we? Just decades in stickleback, localized genomic changes happen on a background of practically no divergence. And that happens pretty much repeatedly anywhere throughout the world, which is crazy to think about. Recently diverged populations have ample opportunities to interbreed when sympatric. And really, this means that we need to think about stickleback as not separate populations, or at one time they were described as different species. There was a species of oceanic stickleback and freshwater. It's really a big metapopulation. And being a metapopulation, that means that the standing genetic variation that is utilized exists in that metapopulation. And much of it has existed for millions of years. So for us to actually really think about, oh, and there's corresponding recombination rate variation across the genome that then is co-varying with those genomic regions that are used over and over again. So if we actually think about this question that I brought up at the beginning, and that why are organisms the way they are, I would posit that we can't actually use for systems like stickleback any specific situation of this oceanic form evolved into this freshwater form, especially if we have the mindset of thinking of sequential fixation, immune mutations, or even just the use of standing genetic variation if that genetic variation is contributing. But we have to take into account the deep evolutionary history of the entire system to be able to say, well, why is it that the freshwater form evolves over and over again? And really leads to the hypothesis that when we say, why does stickleback look like they do, the entire species, we have to take into account the 15 to 20 million year history, and that the adaptively evolved genetic and genomic architecture of stickleback influences the present repeatability and the future evolvability of this species. And what that means is that if we could project ourselves back 15 million years ago, the evolvability itself would be different then than it is now, and that evolvability has actually evolved in the stickleback. And with that, I thank you for listening, and I'm happy to take questions. Before are we doing tea or anything?
Yeah.
All right. Okay. Yeah. So I'm happy to answer questions now, but I'm going to go to tea afterwards as well too, because I haven't had lunch, so I need to eat something. Yeah.
I'm curious if you looked at balancing selection on the for so long and the effects.
Yeah. Yeah, and that paper that from Tom Nelson in there, I didn't show you those data, but we actually have the balancing spatially mediated balancing selection, the patterns at the molecular level that you expect to see in terms of the divergence within versus between is just what you'd expect to see. So that's basically what it looks like, is there's balancing selection of those haplotypes between the habitats. Very much like sex chromosomes, but just not, it's habitat mediated. Yeah, Becca. Yeah, so that's a good, oh yeah, sorry. The question was, are there other organisms that show a similar pattern of divergence all over the world? And I think that I'll take the first part. Are there other organisms that show similar patterns of divergence? There are other organisms that show repeated adaptation to different habitats. And I'll just stick to fish for right now. If you think about this, this is not too different than several salmonid species that actually have freshwater morphs and oceanic morphs. So kokanee salmon are actually a landlocked freshwater morph of sockeye salmon as well too. And there are other situations like that. You know, that's a good question about whether there's any other that show a global pattern like this, though. Being on the spot right now, I would say I bet there are, but they're just not coming to mind at this moment. And when I say global, this is really circumpolar, so the northern hemisphere. So Stickleback all live from about, well, a little bit south, latitude south of us up to the high latitudes, but still it is pretty circumpolar. Andy.
Well, for the term data is if you look at these phenotypes in the GWAS, you and all the groups around the world show small fast signs, mutations, and
yeah.
If you were to go to an EQTS in marine versus freshwater, you find very different distribution of fast. And does it support their hypothesis about there being equivalent to EQTS, or are there just chunks that would be associated with saying a differential topology?
Yeah, things like that. So to repeat Andy's question is that a lot of the work previously that had been done mapping, and then some of that's actually moved into GWAS. And one thing I'll point out is there actually haven't been that many studies actually done in hybrid situations like this. Do you actually, that seems kind of weird. Does that, if you were to do the similar sort of thing using an expression QTL approach, where the phenotypes that you're actually looking at are not morphology phenotypes, but patterns of gene expression across the genome, you see a different pattern? The answer is we haven't done that yet. And so it's a not good aim for a grant proposal to actually do that. So we actually had proposed that in a previous aim. So we don't know the answer to that yet, but the prediction would be that if you have this evolvability, that part of that pattern that you would actually see in those EQTLs is that you actually have a number of cis effects, but you have these long-acting trans effects that are co-localized in those genomic regions that are divergent. Yeah, good question. Yeah.
I was fascinated by the fact you mentioned even can take years to start seeing plant classification, and then that happened with the island years ago,
Yep.
years ago, you already see sort of adaptation to freshwater. I'm curious to know more about the time scales over which you can start seeing plant classification, but over which that rate of adaptation starts slowing down and you're pushing stationarity. So okay, takes 10 years. We start seeing adaptation. When does it reach stationarity?
Yeah, that's a really good question. So to repeat Stilian's question is, to rephrase it, what's actually the shape of the dynamic of the adaptation for that? And I think the answer to that question is we don't really know at this point. It has some sort of shape where it asymptotes. And if you think there are estimates of what R, meaning the field, years ago, it would have been over tens of thousands of years. And then it became 13,000 years, and then it became 50 years from our work and some other. Now with these more recent putting Stickleback into ponds, it's probably on the order of a decade or less, where most of that change seems to happen in those changes in allele frequencies. But I don't think that answers your question, which is, does it asymptote and stop, or does it continue? How much optimization actually goes on after that? Yeah, so it's definitely not linear. It's curvilinear and asymptotes, but whether that's an asymptote too. And if you could do that experiment for millions of years, what kind of optimization would you see? And the reality is many of these systems are ephemeral. And so what is a freshwater population now might not exist in another 1,000 years or even 50 years. Lucas.
Hey, Bill.
Hey, how's it going? Can you ask the question?
Yes. So this is maybe a bit of a philosophical question, but this evolvability is sort of evolving along a certain axis of variation in a sense, right? Like, because there's these sort of repeated acquisitions of the phenotype and it's associated with a specific environment. So do you think, does that generally incur some kind of a trade-off with evolvability in other potentially orthogonal dimensions or that aren't aligned with that axis?
Yeah, that's a great question. Did you all hear that question? Okay, this is a really great question. So if you have the evolution of evolvability of a system that then in some ways channels or canalizes, what are the costs for doing such a thing? And if we put it in the context of phenotypic plasticity, So one potential solution for experiencing a variable environment repeatedly is you evolve phenotypic plasticity. But what happens when the environment changes and you're out of sync there? What are some of the consequences for that? And I think that's an important question in the field, and it's an important question just in general. And I think it's one where we know probably what some of those costs are, but in general, those costs would be incurred for a phenotypically plastic system in a new environment, as well as if those were genetic polymorphism that was segregating, and that was a solution. But there can actually be costs for mismatching of if the environment changes and you incorrectly produce a phenotype that then is maladaptive as well too. So in the Stickleback system, this is just speculation. We don't have an answer to this, but years ago, I used to ask this question, which is, why are there so few Stickleback species? And I actually suspect that it has something to do with it. It's very different than the cichlid radiation, where over roughly the same amount of time, you have thousands of different species. But in Stickleback, we have different axes of the evolution of reproductive isolation, and in a few cases, the evolution of new sex chromosomes. Katie Pickel's lab, June Catano, has shown very nicely that can happen, but we don't have this proliferation of new species. As a matter of fact, we can cross in the laboratory Stickleback from different populations in Alaska and Oregon. So I actually suspect that there may be a trade-off in the higher level evolvability of a lineage in the ability to explore new habitats and new diversification in the very long term. But that's just a complete guess and a philosophical answer to that. Thanks, everybody. This was fun. And this is my first in-person talk in years. I'm happy to do it. What?